Terminal, incurable, fatal. Hearing these words from a doctor means the end of life as you know it. Hearing these words means heartbreaking and insufferable conversations with your friends and your family. However, with advances in molecular biology and stem cell research, scientists hope to make these words less common and less daunting. Before stem cell research, if a patient's liver failed, they had to rely on an organ donor. This solution is less than elegant for two reasons. Firstly, the demand for organs far outweighs the supply. Not only are organs rare, your immune system may reject and attack the transplant like a foreign invader. The first solution presented was the use of embryonic stem cells. When the embryo is developing, these are the first cells to come after the egg is fertilized. These cells can be specialized or differentiated into a subset of other cells, meaning these cells can be used to create other cells, tissues, and organs. While this provides a solution to our problem, we still suffer from the rejection issue. Further, several ethical issues have been posed and the practice itself has been criticized. Induced pluripotent stem cells were first discovered by Dr. Shinya Yamanaka, and this was a huge breakthrough. It solved both issues at the same time. With a lot of research and a little luck, Yamanaka was able to reverse the specialization process and create pluripotent stem cells from specialized adult cells, a process that was thought previously to be impossible. Every cell in the body contains a full copy of its owner's DNA or genome. However, not every cell needs every part of the genome. The part of the genome the cell needs are unwound or expressed, while the genes the cell does not need are tightly wound together and neglected. By inserting a specific set of proteins, a specialized cell can be rewound and reverted to a pluripotent cell. Like embryonic stem cells, iPS cells could potentially solve the organ donor problem. Along with this, the patient's immune system will not attack newly differentiated cells. Granted, the cells are from the patient. The future of iPS cells, while bright, is not quite here yet. The technology is new and comes with several limitations. Firstly, induced pluripotent stem cells are a specialized version of totipotent cells. For example, an iPS cell created from a skin cell cannot be differentiated into a red blood cell. A research article from Nature shows that the first totipotent iPS cells were created just this year, however their mechanisms are not fully understood yet. Another limitation is highlighted in the 2019 issue of the Texas Heart Institute Journal. The author notes that while rebuilding organs is possible, complex organs, such as the heart, are more difficult to build due to their need for electricity to pass through them in a specific manner. However, there is no need to fear. Studies have already shown some progress. For example, one woman fighting progressive blindness was able to halt further degeneration of her eyesight with an iPS cell injection. iPS cells appear to be the future. They trump the shortcomings of their embryonic counterparts while remaining unburdened by controversy. Their mechanisms turn the hands of time at will, and while the science is new, their future seems promising and their potential seemingly limitless.